Thank you very much for the kind introduction. Hello, everybody, and welcome to my presentation. I'm very happy to be here today to talk about continuous manufacturing to you guys. I will talk about continuous manufacturing, the advantages, the challenges of continuous manufacturing, and what is the role of excipients in smooth transition from batch to continuous manufacturing. For today's talk, I concentrate on blending unit operation. First of all, we all know that pharma industry is shifting from batch to continuous manufacturing for many beneficial reasons, such as faster production, reducing the cost. There is no storage. There is no scale-up for these processes. We have better consistency for the product. We have better control over our process and production. And last but not least, we have less footprints with these products. On the right side, I show you the manufacturing side. Unfortunately, you cannot see it so well. At Eli Lilly site in Ireland. So this is the manufacturing site they have. The red line is showing you the batch facility, and the blue line is the continuous facility. So for the same amount of production, installing continuous facility, the size is significantly smaller compared to a batch facility. I emphasize once again for the same amount of production. We believe that future of pharma industry is continuous manufacturing. FDA is actively encourage pharma industry to install more innovation within their lines. There are guidelines that specifically concentrate on supporting, installing continuous manufacturing for pharmaceutical companies in pharmaceutical industry. FDA has approved since 2015, in meanwhile, 7 products to be produced with continuous manufacturing. So Vertex and Johnson & Johnson were the first companies who started, and this year also Pfizer got approval for another product to be produced with continuous manufacturing. So this is what we see, the trend and the market, which direction we are going. But what is exactly the difference between batch and continuous manufacturing? We hear this very often, but I think we should clarify what is exactly the difference. So looking at batch processing and continuous processing, we have this unit operation, but in traditional batch processing, each unit operation is separate. So after each unit operation, the process stops. It is removed from the line, and after quality testing, it is transferred to the next unit operation. So the typical process time for batch manufacturing is in order of months. However, for continuous processing, you see the unit operations are connected. There's no stop in the process, and the product of one unit operation is directly transferred to the next unit operation. The quality testing and quality control is then in line for this process. And the typical process time for this continuous processing is in order of hours or days. So we work closely with a process manufacturer that they have this continuous tableting machine. You can have the device in a room that is significantly smaller than this room that we are now inside. So you put the raw material, API and excipient on one side, and then you get your tablets within 10, 20 minutes on the other side. It's really impressive. So today's presentation is concentrated on the blending. Blenders are have vital role in continuous manufacturing. They should make a homogeneous blend out of API and excipient and deliver it to the tableting device, to the tablet die, to get the homogeneous tablet from the API and excipient. So any inconsistency that we have here. If we have a low concentration of API or high concentration of API, it will be introduced to the next unit operation and finally to the tableting device. So there are many different blenders available on the market. On the right side, you see the batch blenders, and on the left side, you see the continuous blenders available on the market. There have been many studies done on batch blending. We all know that for blending, deagglomeration and effective dispersion are the critical factors to consider for blending, and this depends on the blending strategy. Both filling strategy, if we make layers or if we just put the excipients at once and then the API, and also the blending strategy, what is the blending energy that we use, the blending speed, and also the blending time, of course. The other very important factor is the raw material properties. This defines if we have random mixture or if we have adhesive mixture. This depends on the particle size of the component and the diameter size ratio of API and excipient. This is a very important factor to consider. Many people studied batch blending to understand it. It's very complicated. It also depends on environmental conditioning, the humidity, temperature. There are many factors that impact batch blending, and also the powder bed height in the blender is also a very important factor, as you know. We at DFE Pharma, we also done a study to understand batch blending. We, as an excipient supplier, we look more in details into this. So we, you have the API, and then you should get the excipient that improve the property of your API and also make the processability of your API better. So we looked into different excipients that we have, anhydrous lactose, lactose monohydrate, spray-dried, granulated lactose, and microcrystallized cellulose to grade that we have. And then we made binary mixture of our excipient with a model API, which was paracetamol. And we blended in a turbula blender, and then we took 15 samples, and then we studied the content uniformity of the blend to see what is the impact of excipient that we choose, that they differ in not only in morphology, but also in particle size, bulk and tap density, and therefore flow property, as well as surface area. So we made a different binary mixture also at different API concentration and mixing time, and then we evaluate the blend uniformity with looking at relative standard deviation of the API concentration for every API concentration, as you see, 2 to 30% at different mixing time of 1, 5, 10, and 30 minutes. Now you're looking at data for our anhydrous lactose. As you can see, for low API concentration of 2%, we have a really good blend property for very fast blending. So with 1 minute, we get the lowest RSD. This means that we have a fast deagglomeration for this blend, which can be caused by the smooth surface area of the anhydrous lactose, the high density of this material, as well as good flow property of this material. Interestingly, for the same mixture, with increasing the mixing time, the blending property gets more poor, so RSD increased. Here we think that the segregation tendency, of course, increases, and it is due to the fact that these components, anhydrous lactose and the API that we chose, have different particle size and different density. For the blend with higher API concentration, we see completely the opposite trend. This is very interesting. So for the fast mixing, we have higher RST, and then with the time, the blend content uniformity gets better, RST reduces. This is because adding more API to the blend generally reduce the flow property of the blend, so the blend start to be more cohesive, so we don't have this really nice rolling motion in the blend that help with deagglomerating the API in the blend. But we see with increasing the mixing time, the RST reduces. This is due to the fact that we have more time for deagglomerating, and with the time, deagglomeration happen for this mixture as well. Now looking at our milk lactose monohydrate, for this blend we expected a less segregation tendency, since the particle size of Pharmatose 200M is more or less the same as the Paracetamol that we have. We see that for all API concentration, the intermediate mixing time shows better blend property. However, with increasing the time for 30 minutes, we see that the RST increases, so the agglomeration of API reduces, and this is also due to the fact that these components, they have different in bulk density. For our microcrystalline cellulose, we see a poor content uniformity for some mixtures. For these mixtures, we look in more detail. We have seen that there are several samples that they have higher concentration of API inside them, and that's why the RST increases so much, up to 25%. And this is because we have agglomerated API inside. This is due to the powder property of MCC that we have. So the MCC has generally lower bulk density, and because of that, the agglomeration is slower for this. And we see also at higher mixing time, the agglomeration is happening for this mixture as well. And for this MCC 101, we do not see really a trend related to API concentration or mixing time for batch-wise blending. Okay. I think I'm missing one slide, but it doesn't matter. I show it to you in another slide. I have a summary. So I show you later the data that we have for batch blending of all the excipients that I show you before in the table. So the main question now is, can we really translate all the knowledge that we have from batch blending to continuous blending? For answering this question, first of all, I think we should look into the batch blending versus continuous blending. So for batch blending, we put the excipient and API at once in the blender, and then the blending process starts. However, for continuous blending, as you see here, we have typically more feeders above the blender, and then we are feeding the material continuously into the blender, and then we get our blend. So the process is completely different, and one big benefit of continuous manufacturing is that scale-up is by time and not by size. So all this study that we are doing can be directly used in industry. However, for batch manufacturing, you see the lab scale is completely different than the industrial scale. So there's these all complications from scale-up. For continuous blending, theoretically, we expect less segregation tendency for our mixtures. This is because of the And this is because of the reduced degrees of freedom, so we expect to have less segregation for continuous blender. There is a little study have been done on this. There is one publication from Professor Muzio's team that they compared batch to continuous blending using Avicel and mustard. But we, as an excipient company, we look more in detail. We have done exactly the same study that we did for our batch with a continuous blender, and then we compared the results from batch to continuous for paracetamol with 6 different type of excipient with different properties. So we take from each blend 15 samples, and then we test the content uniformity of the blend and see that how does it differ to the batch blending or not, and if it differs, how. So this is something that is really important to understand, because in continuous manufacturing, the unit operations are all connected, so inconsistency or complication in one unit operation is directly shifted to the next one and affect the functionality and content uniformity of the final drug product directly. So for this study, we compared the continuous blending to the batch blending. So on the right side, you just see the batch blending results for anhydrous lactose that I just showed you before. And on the left side, you see the same data for that we got from the continuous blending. So interestingly, we see that for continuous blending, the content uniformity at all mass fractions, API concentration, and all mixing time, of course time is not so important here, is significantly better than the batch blending. Now I show you the results for all excipients and all mixtures that we tested. So the solid bars are the batch results, and the dashed bars are our continuous blending data. And also I use the same scalar for all the graphs to be comparable. As you can see, for all excipient that we use, the content uniformity for continuous blender is significantly better than the one that we got via batch blending, which is not the case for batch blending. So this emphasizes the importance of powder properties on blend uniformity in batch-wise blending, and this is less relevant for continuous blending. However, we see an exception for our super TAP 11SD for our spray-dried lactose. For this, we have seen that looking at continuous blending results and compare it to the batch, we have seen that here it seems like that batch-wise blending is better and more uniform and more homogeneous blend we get from batch-wise blending compared to the continuous blending. So we have a hypothesis that what can be the reason. So we look into the material properties. We have seen that our spray-dried lactose has generally lower surface area. It means that the particle-particle collisions is reduced for this blend, and also generally it has better flow property, which can lead to better content uniformity for batch, and a bit worse for the continuous blending. So now we come to my last slide. The conclusion that we can get from the slides that I showed to you is, first of all, the material properties, in particular, excipient properties, does affect both batch and continuous blending. And the impact of material properties does differ for these two blending processes. For batch blending, we have seen that we have the best content uniformity for the excipient for our spray-dried lactose. And the content uniformity is relevant to material properties as well as blending time. However, for our continuous blending, we have seen that homogeneity of the blend is less relevant to powder properties. This is a big benefit for our continuous blending and transition from batch to continuous. So this is a good news. So for choosing the right material for continuous blending, we can concentrate on other factors. For example, we can concentrate on to get better compressibility and better functionality of the material, because it is less relevant for continuous blending. However, in continuous manufacturing, as you all know, the unit operations are connected. So if we have an excipient that does not have a good flow and the feeding performance is not good, so we will not get the material inside the blend. It doesn't matter how good the blender is blending. You can blend whatever you get in. If you have inconsistency from the feeder, you get more API, or you cannot get the API in the blender, so you cannot get the good content uniformity as you wish. At the end, I would like to give you a short introduction to our company. We are not only providing excipients, we provide our customers with excipient solutions. We have years of experience and a lot of knowledge on excipients and excipient properties, how you should use it for your process. We work closely with machinery and process suppliers such as Gericke that was contributed to this work, such as Bolle, GA, and other companies to get understanding of how the excipient and the properties of excipient affect the process and, in particular, continuous manufacturing. What else we offer is that we, especially it is relevant for continuous manufacturing, we help the customers to de-risk their use of our excipients. So we help them to understand the variability that is caused by the excipients and what that variability means for the process, which part of the variability is relevant for the process, and how we can control it to get the best quality drug product. So it is important to understand the material properties and the process together to get the best quality drug product at the end. And the other thing that we also provide our customers, our support to install continuous manufacturing within their lines. So we help them to transit from batch to continuous as fast and as smooth as possible. At the end, I would like to thank my colleagues at DFE Pharma, as well as Gericke Group for the contribution to this work. So please do reach out to us if you have any questions on the product, if you have questions on your process, if you need samples, if anything that you need, please do reach out to us. Feel free to reach out, and you can also visit our website here. I'll be happy to answer your question. Thank you very much for your